Hi, everybody. Welcome to today's webinar. Thank you for joining us and spending this 30 minutes to an hour with us on a very special and interesting topic. My name is Jing Li. I'm the Director of Life Science and Technology at GEM Pharmatech US. Today, my great pleasure to have Haibin, Dr. Ruan, with us. Here's a brief intro of Dr. Ruan to share with everybody. Dr. Ruan is an associate professor in the Department of Integrative Biology and Physiology at the University of Minnesota. He's also a faculty at the Center of Immunology. He has a broad scientific background and interest in genetics, metabolism, immunology, and physiology. Having received his PhD from Nanjing University, his thesis work was mentored by Dr. Xiang Gao, the founder of GEM Pharmatech. Haibin's work during this time culminated in a high-profile publication in Nature Communications, researching on the role of stem cell factor KIT signaling in myeloproliferative disease and energy metabolism. Haibin moved on to his postdoctoral research at Yale University with Dr. Xiaoyong Yang. His work established the essential function of O-GlcNAc as a nutrient sensor in glucagon-induced autophagy, hepatic glucose metabolism, and neuronal regulation of beige adipocyte function. After establishing his independent lab at University of Minnesota in 2016, Haibin has been focusing his research activities on understanding how environmental cues and intrinsic signals are joining force to regulate metabolic processes in health and disease. He's taking an integrated approach in studying tissue adaptation and remodeling upon metabolic stress in 4 major areas: O-GlcNAcylation in physiology and disease, adipocyte remodeling and energy balance, intestinal epithelium and gut microbiome, and finally, immune homeostasis and systemic metabolism. These studies have broad implication in obesity, diabetes, inflammation, and aging. Today, it's our great pleasure to have Haibin sharing his research with us. And for today, his topic will be focusing on O-GlcNAcylation and its function at the crossroads of immunity and metabolism. Haibin, without further ado, the floor is yours.
All right. Can you hear me okay, Jane?
Yes.
Okay, cool. Sounds good. Thank you, Jane, again for the invitation and the very kind introduction. So today, I'm going to speak to you about our current research about protein O-GlcNAcylation and in immune response. As Jean just mentioned, my lab really has broad interest in cell-cell communication in both metabolic and immune systems. I have long-term interest in obesity and adipose tissue, so we are still having a heavy lift on this project. Currently studying adipose tissue remodeling in obesity and aging at different locations of our body. Since my postdoc training and established my own lab at the University of Minnesota, I have been always interested in this protein O-GlcNAcylation modification in metabolism and immunity. So currently in the lab, we really use intestine as a model to investigate host and microbe interactions. In addition to protein O-GlcNAcylation, we are also interested in other metabolic pathways in that context. So today I'm going to talk about protein O-GlcNAcylation in immune responses. Excuse me. So whenever you heard the sweet word glycosylation, you've probably been thinking about these structures. As alluded here, on the left are O-linked glycosylations, on the right are N-linked glycosylations. Those are traditional biochemistry textbook structures about glycosylation that modifies extracellular and secreted proteins. As you can see that those different color-coded square or circles indicates different sugar modifications and their derivatives. And I'll note this blue square, which is of course my favorite, is N-acetylglucosamine or O-GlcNAc. As you can see that those modifications are play an important role in structural functions of specific proteins, and in most cases we think these modifications are stable. However, today I'm gonna introduce you a different kind of glycosylation, which is called O-GlcNAcylation. That, in contrast to those two modifications I just mentioned, this O-GlcNAc modification happens inside of the cells on intracellular proteins, and this modification only has one sugar moiety, which is this blue square O-GlcNAcylation on it. So what is this O-linked N-acetylglucosamine? As I just alluded, this is a glucosamine through O-link adding to the serine and threonine of intracellular proteins. Interesting enough, there are only one pair of proteins are involved in the process of these modifications. One is O-GlcNAc transferase, or OGT, adding the sugar to the proteins. On the other hand, O-GlcNAcase, or OGA, removing the moiety from the modified proteins. As I just want to emphasize one more time that this modification is just a single O-GlcNAc moiety on intracellular proteins, and these modification, on and off modifications, can be very dynamic and quickly. A lot of labs in the world, including ours, are interested in these modifications largely because this modification has been indicated as a nutrient sensor. What do I mean by that? So as you could see over here, these the substrate for this modification is called UDP-GlcNAc. It's actually derived a pathway called hexosamine biosynthetic pathway. As you can probably appreciate that the glucose, in addition to used for glycogen synthesis or glycolysis, a portion of that intracellular glucose can shut into this HPP pathway, and also integrating other molecules, including glutamine, acetyl-CoA, and UDP, to finally synthesize this UDP-GlcNAc. So as you probably can imagine, the levels of intracellular UDP-GlcNAc levels are very dependent on the availability of glucose, glutamine, and other nutrients. Since its discovery in 1980s by Jerry Hart at Johns Hopkins, then and now at UGA, our field, even though still small, but has shown a rapid growth over the years. Not just by the publications, but also by the numbers of O-GlcNAc modified proteins. As you could probably appreciate at the bottom right, in humans and in mice, totaling more than 10,000 of genes, proteins, I should say, has shown to be O-GlcNAc modified. And if you're interested, feel free to go to this website and check your favorite protein of interest, whether they are modified or not. So through the years, this O-GlcNAc modification has been implicated in many human diseases, such as cancer, metabolic diseases, aging, and others. So in addition to function as a nutrient sensor, we and others also found that O-GlcNAcylation can respond to many other signaling stimuli, such as stress. We found that starvation and in some cases oxidative stresses can induce O-GlcNAcylation in cells such as liver. Dr. Xiaoyang Yan and others have also shown that circadian is one of the important regulator of protein O-GlcNAcylation in many tissues. In my own lab, we previously found that in the gut, microbiome is actually a determinant of how much protein O-GlcNAcylation can be formed in the intestine. So as So as I just listed over here, inside a particular cell, this O-GlcNAcylation can, in responding to these different stimuli, and regulate a plethora of functions within the cell. For example, crosstalk with other post-translational modifications affecting protein stability and their localization. They can, of course, also regulate protein-protein interaction and their enzymatic activities. I just want to emphasize that this modification, just as any other post-translational modifications, for example, phosphorylation, has context-dependent functions. So you really have to really study individual protein, individual O-GlcNAc modified sites individually. So as I just alluded earlier, my lab is really interested in microbe-host interactions. For many of the in the audience probably can all appreciate that upon an infection, our host cells rely on these pattern recognition receptors to recognize those microbe or pathogen-associated molecular patterns in order to elicit a proper immune responses. However, on the other hand, pathogens can also hijack in the host cell and use their cellular machineries for the dissemination of these pathogens. And in many cases, this interaction is actually mediated through post-translational modifications, such as glycosylation of these viral proteins, such as ubiquination and acetylation of host proteins. So when I established my lab around 6 or 7 years ago now, I just asked a simple question: can O-GlcNAcylation is also involved in microbe-host interactions? As a matter of fact, this actually notion is already known. Not long after the discovery of the O-GlcNAcylation by Jerry Hart, his group also showing that in T cells, upon acute activation of the T cell receptor, you could see a transient but robust induction of protein O-GlcNAcylation, particularly inside the nucleus, as shown over here, suggesting an important role in transcription. Not only the protein modification, but also UDP glucanate levels, the substrate for this modification, also increased upon T cell activation. This is another more recent studies in naive T cells. Activation of TCR can induce protein O-GlcNAcylations. As a matter of fact, until recent, there are more than 1,000 O-GlcNAc modified proteins has been identified in activated T cells. Most of these proteins are involved in TCR signaling, RNA metabolism, processing, and also transcription. So this of course, O-GlcNAcylation is important for T cells. However, T cells does not come with just one flavor, as many of you already know. Depending on the environmental stimuli, depending on what pathogens they are seeing, they can become different subtypes of T cells. For example, Th1 here showing upon interferon gamma and stimulus, particularly at the conditions of viral or intracellular bacterial infections, they can become Th1 cells. On the other hand, Th2 cells are activated upon helminth infection and allergic responses. Th17 cells are responsible for extracellular bacteria and some fungal immunity as well. Interestingly, these so-called regulatory T cells are a group of immune suppressive cells. They express these lineage-defining transcription factor FOXP3 has been showing not only mediating peripheral tolerance of immunity, but also its overactivation has been shown in cancers. Its loss of function has associated with many metabolic diseases. So, however, whether O-GlcNAc modification is involved in Treg cell function was not known about 5 years ago or so. So at that time, my former postdoc being Dr. Bing Liu and a graduate student, Oscar and Christine Hallquist lab at University of Minnesota, they decided to team together to really tackle this question. So we first just stimulate these Treg cells with CD3 and CD28, basically active TCR. As you can expect, just like naive T cells, Treg cells also, upon activation, upregulate protein O-GlcNAcylation. And to determine the functional impact of these OGT in Treg cells, we specifically knock out the OGT gene in FOXP3 expressing cells. Surprisingly, these knockout mice all developed this typical scurvy phenotype in very young age. This is these copies when you don't have FOXP3 gene or don't have Treg cells, suggesting that OGT is really essential for Treg cell identity. When we take took these Treg cells out of the lymph nodes or spleen, we don't see a difference in their total numbers. But one interesting phenotype we saw is that many of these Treg cells actually lose the FOXP3 expression in them. These cells are becoming these so-called EX Treg cells that are actually pro-inflammatory rather than immunosuppressive. Interestingly enough, we found that the FOXP3 protein itself can be O-GlcNAc modified. When you overexpress OGT, you could see a much stronger reduction of the FOXP3 glycosylation, which can be suppressed when you express the O-GlcNAcase in here. So with the help with L. Blingame's lab at UCSF, we were able to identify 5 novel O-GlcNAc sites on FOXP3, and we then mutate them to alanine, and then put them back to Treg cells, and found that the FOXP3 protein compared to the wild type protein show increased, reduced levels, and with the data not shown here, we found that it's due to increased ubiquitination and protein degradation. So O-GlcNAcylation not just stabilize FOXP3 protein, but also the Treg cell identity. Now, in addition to Treg cell identity, we also look at its function. So here, I just show you some of these effective Treg cell markers. For example, on the left, showing these CD44 positive and CD60L negative effective Treg cell populations, they were reduced in knockout mice. Similarly, if you use KLRG1 to label these, you could also see similar reductions. Many studies previously have identified this IL-2 signaling. IL-2 and its downstream STAT6 are not only important for Treg cell development, but essentially indispensable for effective Treg cell differentiation. So when we treat these Treg cells with IL-2 only in YFP negative Y-type cells, they show increased effective marker gene expression, but that was totally ablated in the YFP positive OGT deficient Treg cells. Excuse me. We also, sorting these both positive and negative deficient Treg cells out of the mice, and then did a bulk RNA sequencing. You could see that the most downregulated genes are actually STAT5 target genes. Some of these are shown here. So we, interestingly enough, STAT5 itself can be O-GlcNAc modified. There's one site has been reported on STAT5. So we went on to mutate it to alanine. As shown on the right, you could appreciate that only the wild type, but not the O-GlcNAc deficient STAT5, was able to induce target genes such as SOX1 and SOX3 expression. So OGT modifies STAT5 and it is required for STAT5 activity. Now, in order to really confirm this, really STAT5 is a functional target of OGT in Treg cells, we collaborate with Michael Ruan at Center for Immunology and really overexpress a constitutive STAT5 only in Treg cells together with this OGT deficiency. As you can appreciate over here, this curvy phenotype and enlarge of the spleen and lymph nodes was mostly rescued when STAT5 is overexpressed. But that's not sufficient to totally block the death of the mice, but you can appreciate their lifespan was dramatically extended. Not just these phenotypes you could see, but also if you isolate these FOXP3 T cells, you could find there that the protein of FOXP3 protein expression can be also regulated. Not just the FOXP3, but also effector Treg cell markers can be at least partially rescued. As I just mentioned earlier, loss of OGT will lead to the emergence of these so-called EX Treg cells, but that was prevented if STAT5 was overexpressed. So from these data, we kind of conclude that inside the Treg cells, OGT can target both FOXP3 and STAT5. And the glycosylation of FOXP3 stabilize FOXP3 and leads to the stabilization of the Treg cell identity. Parallelly, the STAT5 glycosylation can ensure the activation of STAT5, which is important for the suppressive function of Treg cells. We still don't have a full picture of how Treg cell TCR activation can activate OGT, but we have some evidence suggesting that calcium and chymogenic kinase are involved in this, and we are currently working on this aspect. So, basically, going back to these different flavors of T helper cells, our study in 2019 showing that OGT is really required for Treg cell function. And then about the same time, there's another group at KU. They actually found that OGT can promote Th17 cell differentiation as well. As some of you may know that Th1 and Th17 has basically counteract each other's development. So they're suggesting that these OGT can suppress Th1 cell development. However, there's a still open question to us is that whether Th2 immune response can also regulated by protein O-GlcNAcylation. As many of you know that Th2 type 2 immune response are basically responsible for expulsion of helminths, parasitic helminths, and also involved in allergic reactions. As listed here, these activation really require two tiers of reactions. The first tier is tier cytokines, is so-called IL-25 and IL-33. But identification, we still don't know really how our host can sense these helminth expressions. I think one reason, I'm going to skip this, I think one of the reason is basically because when in terms of bacteria and viruses, our host cells, particularly macrophage and other innate cells, can use this pattern recognition receptor to recognize structure features of these microbes. Could be, for example, LPS. However, in contrast, in those helminths and allergens, they don't have any, there's no specific structure features can be recognized by our immune system. A lot of times, our immune cells are actually detecting tissue damage and other functional abnormalities associated with infections. So that's why it's hard to identify the sensors of these anti-helminth responses. I just going back to these models again. Thanks to Akiko and Mazadov for making these slides. But as you can appreciate that, again, these Th2 cells can secrete so-called type 2 cytokines such as IL-4 and IL-13. They acting on downstream effector cells such as goblet cells and master cells. And all these cells, together with all these cytokines, can initiate these so-called whip and sweep responses to basically ultimately leading to the expulsion of helminths. However, rather compared to this well-documented downstream effect, the upstream sensing of the helminths and also secretion of these first-order cytokines, including IL-25 and IL-33, are less characterized. For example, in about 6 years ago, we still don't know where this IL-25 was coming from. So we were interested in that. We kind of asked us a bold hypothesis would be OGT and protein O-GlcNAcylation is involved in the sensing or recognition of helminth infections. So about the time I just started my own lab and I was studying OGT in the intestine, 3 groups really published at the same time, and I identified that these tough cells are sole sources of intestinal IL-25. As you can appreciate that in the naive, non-infected wild-type mice, you basically can see only 1 or 2 tough cells per villi crypt unit. And these tough cells has a bundle of microvilli extending to the lumen of the gut. As you can probably imagine, these kind of sentinels of the intestine can kind of survey the environment all the time. When you infect these mice with helminths, you could see a rapid and massive hyperplasia of these tough cells showing in yellow here, and they secrete a lot of IL-25. So the current knowledge is that when facing a helminth, still we don't know the sensing mechanism yet, but tough cells can secrete IL-25, and these IL-25 cytokines can act on both innate and adaptive type 2 immune cells, and those once activated, these type 2 immune cells secrete type 2 cytokines including IL-13 and IL-4, and those cytokines can act on not just the intestine but many others, but in terms of intestine, they can act on these progenitor cells and tell the intestine to make more tough cells and at the same time goblet cells. So basically this forms a feed-forward loop, and these goblet cells can secrete mucin, and it's very important for the whip and sweep response. Now, my former postdoc Min Zhao and former visiting scholar Kai Chunren, when they joined the lab, they decided to team up to ask a question whether OGT and protein O-GlcNAcylation is involved in these feed-forward whip and sweep responses. So in order to study this, we really need to find a murine helminth model. And in this case, we use this H. polygyrus. I will not spend time to really describe the lifestyle of these helminths, but we got great help from Jakob at Washington University. He was kind enough to really taught us how to use those worms and analyze them. One of the first things we initially found is that if you just infect these wild type black 6 mice with H. polygyrus, you could see a rapid but also sustained sustained strong activation of protein O-GlcNAcylation in the intestine. This induction is parallel to the tyrosine phosphorylation of STAT6. I probably forgot to mention STAT6 is basically downstream of the type 2 cytokine IL-13 and IL-4. STAT6 is absolutely required for type 2 immune response. Without STAT6, there's no tough cells, there's no efficient whip and sweep responses. Now, because of these findings, we ask ourselves, okay, maybe OGD is required for this anti-helminth response. So what we did is using Vinein-3 to specifically delete OGD gene in the intestinal epithelial cells, and then infected these mice with H. polygyrus. On the left, you can see that in the wild-type mice, there's a massive amplification, differentiation of those DCLK1 positive tough cells. However, that was totally abolished when OGD was deficient. The quantification of tough cell numbers is shown here. As a result of the loss of tough cells, you're going to lose IL-25 secretion from inside of the intestine. Remember, IL-25 is only secreted from tough cells. As a result of defective IL-25, mice cannot efficiently clear helminths from the intestine. So as a result, you could count much more eggs inside of the feces. In addition to H. polygyrus, we also use a different worm called Nepo-strongylis, which is it's more kind of rat, natural rat helminths, but can also infect the mice, but can be rapidly cleared in mice. So we just use this to really confirm different this response is not specific to just one type of worms. As you can appreciate over here, basically tough cell hyperplasia, IL-25 expression, and worm clearance was all defective in OGD deficient mice. As I said, type 2 immune cells are essential for the response, the anti-helminth response. So we went on to characterize those cells. As you can appreciate here, Th2 cell numbers was reduced. ILC2 proliferation was severely impaired. As a result of that, type 2 cytokines, including IL-13 and IL-4, was significantly downregulated. These IL-4 and IL-13 can activate eosinophils. That number in knockout mice was dramatically reduced. Goblet cells are increased in number and size upon type 2 cytokine stimulation. However, that response was impaired when OGD was not there. So with this data suggesting that really OGT is important for this tough cell differentiation and successful mounting of the anti-helminth response. However, we really don't know, even though we use the intestine-specific knockout, we still don't know where OGD is acting. In the progenitor cells or in the tough cells, in goblet cells, or any other subtypes of the intestinal epithelial cells. Also, knocking out OGT in Vinein-3 can lead to sustained inflammation that can confounding our interpretation of the data. So we decide to use an alternative approach to prove this. So my postdoc, Dr. Xiaoyou Yan, pictured here, he was kind enough to give us these ROSA26 Cre-dependent overexpression of OGT mice. And when we crossed to Vinein-3 to only overexpress OGT in the intestine, interesting enough, and it's very gratifying to see that both at the naive conditions and after helminth infection, overexpression of OGT itself was sufficient to increase tough cell numbers in the intestine. We also did the opposite approach to overexpress the O-GlcNAcase inside of the intestine. OGT overexpression leads to reduced tough cell marker genes such as DCLK1 and POE-TREG3. So then we asked whether this action was because of OGT in the progenitor cells or mature cells. The first thing we did using the OLFM4 CRE-ER to delete OGT only specifically in the progenitor cells. This was done in Dr. Shivan Shon's lab at Xinjiang Medical University. Upon tamoxifen induction of the knockout, we could see that there's a reduction of IL-33 induced tough cell hyperplasia. On the other hand, when we knock out OGT specifically only in tough cells using this DCLK1 CRE-ER, we did not see any changes in tough cell numbers or responses to a Helminth infection, suggesting that OGT is important during the differentiation of the tough cells, but not so much in the mature tough cells. As I just mentioned that STAT6 is essential for tough cell development. So the question was whether OGT is actually targeting STAT6 to regulate tough cell differentiation. So in intestinal organoids, when we treat IL-4, you could see a STAT6 phosphorylation, but that phosphorylation was reduced when you co-treat the cells with two structurally different OGT inhibitors. In vivo, when you compare wild-type versus knockout intestine epithelial cells, isolated pure intestine epithelial cells, you could see a reduction of tyrosine phosphorylation of STAT6 upon Helminth infection. Interestingly, STAT6 protein itself can be O-GlcNAc modified, showing over here after IP. This signal can be enhanced by OGT co-expression. Most importantly, when we test using luciferase assay to test the STAT6 activity, we found that only the wild type, showing in black, but not the catalytic dead OGT, showing in gray, was able to induce STAT6 activation. Also similarly, IL-4 activation IL-4 cytokine treatment can increase STAT6 and transcription activity, but that can be ablated if we treat it with an OGT inhibitor. So in order to really test STAT6 O-GlcNAcylation is functionally important, again, we seek help from Dr. Al Burlingame at UCSF. We sent the purified STAT6 to their group and they did mass spec, and I was able to identify at least 8 O-GlcNAc sites in the C-terminal of the STAT6. These 8 sites actually cluster into 3 groups. One cluster is close to the tyrosine phosphorylation site. The other 2 more C-terminal sites are actually in the transactivation domain. So we mutate individual sites or in groups. As you can see over here, these 2 sites, mutant 2 and mutant 3, show dramatic reduction in glycosylation levels. And if you mutate them together, donated as mutant TAD, you barely could see any O-GlcNAc levels remained in this STAT6. As a result, when you do a luciferase assay, you can see that compared to wild type, showing in black here, all these mutants, particularly this mutant TAD, show a severe reduction in the ability of inducing STAT6 transcription. As I probably mentioned earlier, that this POU12F3 is a transcription factor that defining the tough cell lineage. Its regulation was not known. And in here, we found that STAT6 actually is basically the master regulator of POU12F3 transcription, showing here, and that activity is dependent on glycosylation because mutating glycosylation on this TAD domain ablated the transcription activity of STAT6 on POU12F3 promoters. This is not just happening in cell culture, but also in organoid cultures, where you can introduce wild type versus mutant STAT6 into organoids, and then differentiation to be tough cells. And we can see that tough cell markers was reduced when STAT6 cannot be O-GlcNAc modified. So we always want to be thorough to really see whether STAT6 is really important for the OGD function, because as I just mentioned, there are thousands of protein can be O-GlcNAc modified. How did you know this specific protein is responsible for all the phenotypes you've seen here? So in order to test this, we collaborate with Shiwen Shon again at Xinjiang Medical University, and also with the help from Gem Pharmatech. Is actually they help us to generate these Vinencre dependent constitutive active STAT6 overexpression mice. So we have at least 3 lines being generated there, and what we did is cross these intestinal specific STAT6 overexpression mice to OGT deficient mice. As you can see, appreciated over here, both using DCR-CoR1 to stain tough cells or use Elsing blue to stain goblet cells, you could see that the both tough cell and goblet cell numbers can be at least partially rescued by overexpress these constitutive active STAT6. So suggesting that STAT6 is a bona fide target and a functionally important target of OGD in tough cell lineage. So from the data I just showed you so far, I think we can conclude that OGT modifies STAT6 and that modification is really required for STAT6 to activate PUU12-3 and tough cell differentiation, and IL-25 can in turn activate type 2 immune cells. However, at that time, we were wondering, probably IL-25 cannot explain all the phenotypes, and also IL-33 is another alarmin, so-called alarmin that's secreted by epithelial cells and then instruct these type 2 cytokines. Their secretion was not known. So we decided to test this hypothesis as well. So the first thing we did is actually did just the RNA sequencing, comparing OGT knockout versus wild type. As you can see that those proteins called gastamine C genes, gastamine C2, C3, and C4 are the most downregulated genes in OGT knockout epithelial cells. On the other hand, if you treat organoids with IL-4 to activate the type 2 signaling, gastamine C2 to 4 genes were the most upregulated. So what are these gastamines? Many of you probably heard gastamine D and gastamine B. So this is a group of gastamine genes that are very important in innate immune responses. As you listed here, all these family proteins has N terminus and C terminus. When they are linked together, they are inactive. Once they are activated by caspase and other proteinase, the release of the N terminal of the protein can form in pores on the cell membrane. So the gastamine D probably is the most studied member of this family. And for example, inside of the macrophages, upon activation of the inflammasome, gastamine D can be cleaved by caspase 1 or caspase 11. The release of the N-terminal can form pores on the cell membrane. That pore formation can at least do 2 things. First is mediating the release of pro-inflammatory cytokine, including IL-1 beta and IL-18. Remember, IL-1 beta and IL-18 are type IL-1 family cytokines. They do not have signal peptides, so they have to really rely on non-conventional ways of secretion. And this pore formation is basic one of them. When these pores cannot be repaired and forms too many on the cell membrane, it could eventually lead to a specialized form of cell death called pyroptosis. So because we found gastamine C is a target of OGT. However, the function of GSDMC compared to many other family genes was not known at that time. So we decided to test this in helminth infection and found that GSDMC was the only one can be induced by helminth infection compared to other family genes. Also importantly, the GSDMC can be cleaved upon helminth infection. STAT6 is absolutely required for GSDMC and upregulation by IL-4 cytokines. Most importantly, to confirm the RNA sequencing data, we found that OGT overexpression can increase the transcription of GSDMC C2 here. On the other hand, overexpression of OGA or inhibiting OGT can suppress its transcription. We know this regulation is STAT6 dependent because only Y type but not the O-GlcNAc mutant STAT6 can fully activate GSDMC luciferase assay. This is not just in cells but also in organoids. As you can see here, endogenous GSDMC gene levels is dependent on STAT6 glycosylation. So we want to know whether GSDMC is really functionally important. With the help of Dr. Shango, the founder of the Gem Pharmatech, he was kind enough to show us these mice, and we generate them, get them, and make a Wheating-Cree specific knockouts, and infect them with helminths. And as you can appreciate over here, these tough cell markers and tough cell numbers is reduced. Goblet cell numbers is reduced. On the other hand, you could see more helminth eggs in the feces, suggesting that GSDMC is important for is required for helminth expulsion. However, we spent really quite a lot of time to try to figure out the mechanism. We did RNA sequencing, we did culture experiments, isolating cells, and we basically couldn't find any changes if we just comparing Y-type versus knockout mice. For example, here gene sequence, you could see that there's just less than 50 genes differently expressed between Y-type and knockout. So, so with this data, we kind of hypothesize that maybe GSDMC is not functioning cell autonomously. It is acting through a cell non-autonomous pathway to regulate helminth infection. So maybe it really analogous to macrophages and GSDMD, the GSDMC can also be cleaved upon helminth infection, and that pore formation may mediate the IL-33 secretion from intestinal epithelial cells. I just want to remind you and emphasize that IL-33 is also an IL-1 family protein. It also lacks the signal peptides, and it has to be released through non-conventional pathways. So we found that indeed intestinal epithelial cells, particularly these goblet cells marked as AG2 here, and also Paneth cells in the intestinal epithelial cells, express IL-33. GSDMC protein, here just showing RNA, is mostly expressed at the bottom of the crypt, or these so-called transition amplifying cells. However, if you look at the protein, it is not expressed in not expressed at the crypt, but at the villi of the intestine. This is actually a kind of well-defined phenomena in the intestine. There's a disorientation between mRNA and protein expression. Nonetheless, you could really appreciate that this GSDMC, particularly after helminth infection, they localize to the cell membrane on the small intestine. So it can be a conduit for IL-33 secretion. Indeed, that's the case. Inside of the cells, you could see that induction of GSDMC pore formation can increase the medium secretion of IL-33. We know that this is not pyrolysis dependent, because if you're providing glycine, this still happens. Most importantly, if you're getting these epithelial cells, primary epithelial cells from the mice, you could see that the IL-33 secretion to the medium was ablated if we don't have OGT. that can be measured either by Western or by ELISA. This is functionally important because we know if we provide mice with exogenous IL-33, here showing in red, this defect in terms of export the worms is really normalized compared to wild type here. If I have time, I maybe just go through the last piece of data. So the data so far really showing that GSDMC is important for IL-33 secretion. So IL-33 is a cytokine not just only important for type 2 immune responses, but its overactivation can also lead to intestine inflammation. So we wonder, okay, maybe GSDMC, in addition to type 2 immune response, maybe also is involved in intestine inflammation. So what we actually just did 2 simple experiments with help from Dr. Ninja from Nanjing University as well. We found that IL-10 knockout mice, which is a spontaneous colitis model, you could appreciate that GSDMC protein expression and the cleavage is induced. And when you do a double knockout, and you could see that the weight loss in IL-10 knockout mice can be rescued if you GSDMC was knocked out at the same time. We also use a different model where you treat the mice with a chemical called DSS in the drinking water. Upon that chemical induction, you could see that GSDMC protein and cleavage also induced. Interesting enough, if we use H. pylori to induce GSDMC expression, and that can exacerbate colitis in wild-type mice, showing in the black here. But if we knock out GSDMC in the intestine specifically, you could see that can be partially rescued. So suggesting that GSDMC mediated IL-33 secretion may also promote intestine inflammation. So with all that, we kind of conclude these findings in this graph. Basically, we found that O-GlcNAc can O-GlcNAc modify STAT6 upon helminth infection. The exact signal for that was not known yet. We are working on this. The activation of the glycosylation of STAT6 can at least do 2 things. First, it can increase the transcription of POU12 and top cell differentiation, which is going to lead to increased IL-25 secretion into the lamina propria. Second, glycosylation of STAT6 can increase the transcription of GSDMC, which can form in pores and on cells such as goblet cells to increase IL-33 secretion. Together, these type 2 immune cytokine milieu is really important for these whip and sweep responses. So with that, I just like to thank everyone being involved in this study. I think I mentioned most of the people's name when referring to specific data. I want a particular shout out to Minzhao and Kaichen, who is the driving force for this type 2 immune studies. I'm very grateful for all the collaborators that I have currently and previously, and for particularly my mentors such as Xiang Gao and Xiaoyong Yang. I'd like to thank my funding from NIH and also American Diabetes Association. And with that, I'd like to take any questions you might have. Thank you.
Well, thanks, Haibin. That's a densely packed data presentation. Thank you so much for sharing the science and your research. Yeah, I'm going to look into the Q&A. For anybody who have a question, we have 4 minutes up to the hour to address. I'll read it out to Haibin if you have put in something there. As I can see, there's one. I'm going to read it to you, Haibin. Did OGT deficient Tregs lose FOXP3 due to FOXP3 protein degradation? Is it related to other post-translational modifications of FOXP3, such as phosphorylation or ubiquitination?
Yeah, that's a great question. We have not looked at phosphorylation, but we did look at ubiquitination. So what we found is that O-GlcNAcylation on FOXP3 can suppress ubiquitination, polyubiquitination of STAT6, and that thus leads to the stabilization of the FOXP3 protein. Yeah, that's a
Okay.
good question.
I'm gonna read another one to you. I think that's the only time we have left for addressing one more. So the question is, the feed-forward network of tough cell differentiation from progenitor cells is super interesting. Do you have insights on how this feed-forward network prevent itself from oversaturating and leading to damage of the tissue itself?
Oh yeah, that's a great question. I think the field don't have a good answer to that yet. I think there, at least to my knowledge, helminth itself. This is just one part of the story, right? There are negative breaks on this pathway for sure. There are a few studies showing that helminths actually can secrete certain proteins and can activate on that, can negatively regulate the differentiation of tough cells. I was trying to remember the name, but I'm sorry, I couldn't remember the name of that protein. It's a helminth protein, by the way. And
Okay.
also, there's data suggesting that the interferon gamma signaling may have a negative role here as well. But yeah, this is a good question, and we'd like to learn that more as well in the
Okay.
future.
Well, yeah.
Yeah.
Well, thank you so much. There are two more questions. However, due to the time, I cannot address it today. So welcome the audience to email us the questions. We'll pass it on to Dr. Ruan. So thank you again, Dr. Ruan, for sharing your data, your research, and your science. And thanks everybody for joining us today. And we hope to see you in September.
My pleasure. Thank you, Jean, again for the invitation. I will see you around. Have a good
Bye.
one.